Good morning, students. I am meeting you after a very long lapse of time to discuss the lesson, to continue the lesson, that is body fluids and circulation. Recall that one, what I talked about, what I taught you, the composition of blood, the orange factor, the ABO blood group system. Now the next one, the mechanism of coagulation of blood. So when accidentally you are cutting your finger, there is a loss of blood. So this is because of what's called trauma. The meaning for trauma, impact of force or injury. So because of trauma or injury, this is always called what is known as injury, impact of force. So because of trauma, you are making just actually some sort of there is incision on your fingers and on the body. The blood is oozing out continuously. After some time, the bleeding stops. This is because of what? The conversion of liquid blood into a gel-like clot that blocks what we call the opening present at the blood vascular wall due to injury until it is being sealed. Okay, and that is what we call the clotting. It is one of the defense mechanisms against the loss of blood. One of the defense mechanisms against the loss of blood. So what is coagulation of blood? The mechanism by which excessive blood loss is prevented by the formation of gel-like clot is called blood coagulation or blood clotting process. Okay, coagulation or blood clotting process. And here the liquid blood gets converted into a gel-like substance made up of some fibrin threads with blood cells which are entrapped. You'll see later. Now what are the steps involved? Now the clotting process begins when we have the endothelium of the blood vessel. Endothelium is the innermost layer of the blood vessel. So the clotting begins when this endothelium, nothing but cuboidal epithelial cells of the blood vessel is damaged. We have different layers. We are going to see the structure of blood vessels in the next step. Okay, so the endothelium of the blood vessel is damaged and the connective tissue present in the wall of the blood vessel is exposed to the blood. So damage of the endothelium and exposing of the connective tissue of the blood vessel to the blood. As a result, what is happening? The platelets, one of the components of the blood, and here stick to the collagen fibers, adhere to the collagen fibers. The collagen fibers are fibrous proteins found in the connective tissue. So the platelets adhere or attached to or stick to the collagen fibers in the connective tissue and release substances. And as a result, a structure called platelet plug is formed. A plug-like structure made up of platelet and some other substances released, which provides emergency protection to the blood loss. It is not a permanent protection. It provides emergency protection to the blood loss. That's the first step. So damage of endothelium, exposure of connective tissues to the blood, attachment or adhering of the platelets to the collagen fibers of the connective tissue, and release of certain substances. All these things together make up the formation of the platelet plug, which normally closes the opening temporarily. That provides emergency protection to the blood loss. But what is actually happening? So now at the tissue damage or the blood vessel damage, we have release of certain clotting factors from the damaged platelets or from the tissues. And these clotting factors mix with the clotting factors of the plasma. So what's the meaning for that one? Now there are 13 factors are found in the plasma, the major factors, along with some minor factors. Factor 1 to 13. For example, fibrinogen, prothrombin, thrombokinase, calcium ions, that is what's called labile factor, accelerant, proconvertin, and hemophiliac factors. thromboplastin, that is components, that is otherwise called what we have, that is Christmas factor, anti-hemophiliac factor, like that we have so on. Nearly 12 Stewart factor, Hageman factor, fibrinogen stabilizing factor. It's not necessary for. So 13 major factors are found in the plasma. Now the factors released from the damaged platelets and the tissues mix with what we have these factors, either 1 or 2 or many factors, 1 to 30. Now I mentioned about platelets release factors. These factors are called prothrombin activation factor, otherwise called as prothrombin activation factor. The prothrombin activation factor, simply called as thrombokinase, different names are given, released by the damaged platelets, and also that's what we can see, the clumped platelets in the yolk blood, in the platelet blood, and also from what we have the damaged tissues. And these factors, the prothrombin activation factors, mix with the factors present in plasma. So two factors, one group of factors released by the damaged platelets and tissues. Another factor, what is the factor formed in the plasma? They are 1 to 13. Factor 1, 2, 3, like that, up to 13, the Roman letters. Now this is what is happening. And this is followed by the major steps. 1. The conversion of prothrombin to thrombin. 2. The conversion of fibrinogen to fibrin. 3. The formation of what we call a patch, that is a meshwork of fibers. And finally, the formation of clot. Like that, we'll see one by one. So the 3 steps we have to follow. That is what I say. Now, normally, I mentioned about the clotting factor released from the clumped platelets. They are produced by 2 ways, either by intrinsic pathway or by extrinsic pathway. In intrinsic pathway, now the activation factor, the prothrombin activation factor, is produced by the trauma of the blood, by the force damage in the blood. And what we have, the extrinsic pathway, that is the prothrombin activation factor, is produced also by extrinsic pathway, where this is because of the damaged blood vessels. So either because of damaged blood vessels or because of damage to the blood, we have the formation of factors. These factors are called, we can say, that is normally prothrombin activation factor. Now the next step in the clotting process is the conversion of prothrombin, a kind of protein produced in the liver, into active thrombin in the presence of calcium ions and prothrombin activation factor. So the prothrombin activation factor is released by the damaged platelets and the tissues. So here it's not mentioned in the book, but normally the prothrombin gets converted to thrombin in the presence of calcium ions and the prothrombin activation factor. It's nothing but thrombokinase, not mentioned in the book. Now, in the presence of vitamin K, that is given in the book, but vitamin K is essential for the formation of prothrombin. In the liver, prothrombin is formed in the presence of vitamin K. As vitamin K prevents what is called the hemorrhage, it is called anti-hemorrhagic vitamin and promotes the clotting process. So the next step, the protein called prothrombin, I mentioned produced in the liver, is converted to its active form called thrombin, which is nothing but a proteolytic enzyme. It acts as an enzyme in the presence of calcium and vitamin K. So in the presence of calcium and prothrombin activation factor and the vitamin K for the formation of prothrombin in the liver. Now once the thrombin is formed, it acts as a proteolytic enzyme, acts enzymatically on another protein, what is called fibrinogen, a fibrous protein also produced in the liver, into a network of threads called the fibrin threads. Okay, so fibrinogen gets converted into fibrin threads in the presence of thrombin, which is acting as a proteolytic enzyme, acts enzymatically that is fibrin origin to fibrin. To begin with, we have the formation of fibrin monomers. These fibrin monomers undergo polymerization process to form a fibrin polymer that is called actually a soft clot. So it gets converted into a hard clot. Okay, now what's happening? The threads of fibrin interlinked, connected with one another to form a patch that traps the blood cells, okay, and seals the injured blood vessel until the blood vessel is healed. You see that one, this is the region of injury. We have the network of fibrin threads in the form of a meshwork network, and which entraps the blood cells. Here, the red one are nothing but the entrapped blood cells. The black lines represent the fibrin threads. So, a clot is formed of two components. One, the fibrin threads plus blood cells. Maybe I asked one answer. A clot is formed of fibrin threads plus blood cells together form a complete diaphragm-like structure, sealing the opening of the injured blood vessel until the blood vessel is healed. Okay now. So after some time what will happen? The fibrin threads contract and a fluid is oozing out. A yellow colored fluid is oozing out through the meshwork, and that fluid is called serum. Am I done later? So what is serum? What's the difference between serum and plasma? Now in serum, it is different from plasma in not having fibrinogen protein. So plasma without fibrinogen is called serum. That's the main difference. Plasma contains fibrinogen, but the serum contains no fibrinogen, because the fibrinogen is already being used for the formation of the clot. So the fluid that is oozing out through the meshwork after the contraction of these fibers in the form of straw-colored fluid is called as a serum, which contains no fibrinogen. So a plasma without fibrinogen is called what is known as serum or defibrinogenated, another word given in the NEET question paper, defibrinogenated plasma. Defibrinogenated plasma. That's a plasma without what's called fibrin. Now what will happen after the formation of clot? The next step, formation of serum. What is serum? After some time, the fibrin fibrils contract and a straw-colored fluid is oozing out through the meshwork called the serum. So after some time, the fibrin fibrils contract, squeezing out a straw-colored fluid through a meshwork. This is a straw-colored fluid through the meshwork called serum. So a serum is nothing but plasma without the fibrinogen protein, because the fibrinogen is already being utilized for the formation of clot. So it's also called defibrinogenated plasma. The meaning without fibrinogen, plasma without fibrinogen. So serum is equal to plasma minus fibrinogen. That's without fibrinogen. That's called a serum, but it's a straw-colored fluid. Now what is heparin? To maintain the fluid nature of the blood inside the blood vessels, there is a substance which prevents coagulation within the blood vessels. That's called intravascular clotting. That substance is called heparin. As it prevents blood coagulation within the blood vessels and small blood vessels, it is called anticoagulant. Prevents coagulation. This is produced by one type of cells present in the connective tissues, what is called the mast cells of connective tissue. The mast cells also secrete another substance, what is called histamine, involved in allergic reactions. So anyway, heparin is an anticoagulant as it prevents coagulation within the blood vessels, produced by mast cells in small quantities. The mast cells are found in the connective tissue. So it prevents coagulation, I mentioned already, inside the blood vessels and small blood vessels. So intravascular clotting within the blood vessels is prevented, and the fluid nature of the blood is maintained because of heparin. So with that I concluded the coagulation. I will show the diagrammatic or schematic representation of the coagulation of blood in the form of a flow chart. Now this is the schematic representation of blood coagulation in an injured blood vessel. Now we have first the emergency protection for blood loss, a stimulus, that is an injury, which causes tissue damage that results in the formation of what is called the platelet plug. And we have the muscle spasm due to this tissue damage. Muscle spasm means tightening of muscles. If it is undergoing contraction relaxation, there is a bleeding process proceeding. In order to prevent the bleeding process, now the muscles become tightened at the site of injury, and that is called muscle spasm, and leading to the formation of platelet plug formation. That is a collagen fibers. The platelets stick together along with the releasing factors to form just what we have platelet plug, which is an emergency protection. But we have intrinsic clotting mechanism. I mentioned already extrinsic and intrinsic pathways. In extrinsic pathway, we have the damage to the blood vessel. The results in damage to the blood vessel. Intrinsic clotting mechanism that occurs in the blood due to damage to the blood. Now we have the production of prothrombin activated substance from the damaged platelets, and what we have the tissues, the damaged tissues. Now that factor converts prothrombin into thrombin. Conversion of prothrombin into thrombin with the help of calcium ions, vitamin K, and some other factors, particularly the prothrombin activator factor, that is also responsible for converting the prothrombin to thrombin. Now the thrombin acts enzymatically on fibrinogen and converting the fibrinogen into fibrin threads. So fibrinogen forms threads of fibrin. This is because of the activity of thrombin, which is acting as a proteolytic enzyme. Now the fibrin threads at the damaged site of an injured blood vessel traps blood cells and platelets. So fibrin threads at the damaged site of the injured blood vessel traps blood cells and platelets, forming a complete diaphragm, and that is called as a clot, consisting of fibrin threads plus the trapped blood cells. And this is the one which shows what is called the schematic representation of the coagulation of blood in the human body. Now let's see the composition of lymph and its functions. About 90% of fluid that leaks from capillaries eventually seeps back into the capillaries. What about the remaining 10%? The remaining 10% of fluid is collected and returned to the blood system by means of a series of tubules or ducts called the lymphatic vessels or the lymphatics. The fluid inside the lymphatics is called the lymph. Okay, what are the components of the lymphatic system? It mainly consists of three components. Number one, a complex network of thin-walled ducts. They are called the lymphatic vessels. Then filtering bodies. All along the entire length of the lymphatic vessels, here and there, we have small swollen structures called the lymph nodes. They are responsible for filtering the invading microorganisms. They are called as the lymph nodes. Then the third one, a large number of lymphocytic cell concentration in various lymphoid glands. That is the lymphocytes of different types, B lymphocytes, T lymphocytes, etc. Now if you are taking the lymphoid vessels have smooth walls and they run parallel to the blood vessels. Actually, they are found in the skin along with digestive and respiratory tracts. So the lymphoid vessels have smooth walls that run parallel to the blood vessels in the skin, the respiratory tract, and the digestive tract. Now the lymphatic vessels serve as return ducts for the fluids that are continually diffusing out from the blood capillaries into the body tissues, because they are collecting the fluid, returning the fluid to the blood, hence called as a return ducts. So the lymph fluid must pass through the lymph nodes, which are found all along the entire length of the lymphatic system, for filtering the invading microorganisms. That's why it's a must for the lymph to pass through the lymphatic nodes or the lymph nodes, because they filter the lymph fluid, that is, filtering the microorganisms. Now, the lymph nodes that filter the fluid from what we call the lymphatic vessels of the skin. Okay, the lymph nodes, normally you see that one that filter the lymph fluid from the lymphatic vessels of the skin, are concentrated in different parts of the body, but more concentrated in specific regions. For example, the neck region, then the inguinal region. The region below the abdomen is called the inguinal region. Then the axillaries region, that is what we call the armpits region. In the digestive tract, as well as the respiratory tracts. Though we have the lymph nodes formed all along the length of the lymphatic system, they are highly concentrated in specific regions like neck, inguinal, what we call the digestive, respiratory tracts, and also the armpit region, otherwise called the axillaries. Now, what is the course of circulation of lymph? The lymph which is flowing out of the lymph nodes, that is actually after filtering, and it flows into large collecting duct. The large collecting duct normally located in the thoracic region, hence called thoracic lymphatic duct. They are left and right thoracic ducts. So the lymph flowing out of the lymph nodes flows into large collecting duct. They are located in the thoracic region, namely left and right lymphatic duct, or right and left thoracic duct. Now, from the thoracic duct, now the lymph is drained into veins. A large vein located in the shoulder region called the subclavian vein. And from that vein, the blood is emptied into the bloodstream. So, flowing out of the lymph nodes, the large collecting ducts, namely left and right thoracic duct, from the lymphatic duct, the lymph is entering into a large vein present below the collarbone, what is called clavicle, and that is called subclavian vein, otherwise called as the shoulder vein, present below the collarbone, that is the clavicle. From where the lymph is emptied into the bloodstream. That's about the course of circulation of lymph. Now, if you are making a cross section of a lymph node, it shows a number of narrow passages. These narrow passages are called sinusoids that are lined with macrophages. They are nothing but modified monocytes, a type of WBCs. So they are phagocytic in function. That's why you see when the lymph is passing through the lymph node, the microorganisms are phagocytosed by these macrophages. Okay, the cells which are found in the lymphatics, the lymph vessels, or in the lymph, are called lymphocytes. There are two different types of lymphocytes we'll study later. One, the B lymphocytes, and another one, T lymphocytes. Both are involved in immunity. Now, what are the functions of lymph? Now, we have the lymph nodes acting as filtering bodies. The lymph nodes successfully prevent the invading microorganisms from reaching the bloodstream by filtering those microorganisms. So they cannot escape from the lymph nodes. And these microorganisms are phagocytosed by the macrophages. The second one, so lymphocytes, these are all the cells, collect in the lymphatic fluid, are carried via arterial blood and return back to what we call the lymph. Simply we can see the lymph is responsible for the transport of lymphocytes from the lymph glands. The lymph glands or the lymphatic ducts, we can say the lymphatic ducts or the lymph. The lymph is responsible for the transport of lymphocytes from the lymph glands to the bloodstream. Now, the third one, in the intestinal villi, we have a central vessel or the lacteal vessel, otherwise called a lymphatic vessel, surrounded by a network of capillaries. These are the components in the villi region. Now, while the various materials like glucose, amino acids are absorbed into the bloodstream, the fats are absorbed through the lymph formed in the lacteals. The lacteals are nothing but, you see that one, the lymph vessels having the lymph concerned with the absorption of fat. So fats are absorbed through the lymph in the lacteals, the lymphatic vessels present in the building of intestinal wall. So fat absorption needs the lymph, and that occurs through the lacteal vessels, form the unit of what is called absorption, the villus. Now this is the diagram showing the drainage of tissue fluid in the lymph vessel. Now this is the lymph vessel connecting the tissue fluid from the surrounding medium. That is what we can say ECF, extracellular fluid or interstitial fluid, or we can say that simply the lymph. Now the tissue fluid is drained into the lymph vessel. Now we have on the walls of the lymph vessels, walls are present, wall in lymphatic vessel, wall for regulating the flow of lymph. So here and there we have the walls for regulating the flow of lymph into the lymph vessel. Now this is the tissue made up of cells surrounding what we have, the lymph vessel, and we have interstitial spaces also between the cells. Now the collected lymph is carried by the lymph vessel. The lymph vessel, after joining to form a large lymphatic vessel, opens into the subclavian vein, which ultimately is emptied into the bloodstream. With that I concluded the coagulation of blood and the composition of blood and its functions. We have to proceed the next part of the lesson in the next class. Thank you.